oligonucleotides, or ASOs, are a growing class of pharmaceutical drugs that target specific RNAs based on their sequence and cause changes in how those RNAs are used, their processing, their degradation, things like this. They can be designed to target specific, like mutated RNAs, and therefore correct for various diseases like spinal muscular atrophy. So today, let's try to make some sense out of these antisense oligonucleotides.
So what is an antisense oligonucleotide? Well, the name tells you, at least if you've had years of scientific training that have taught you all this jargon. So let me decode it for you. Basically, when DNA is double-stranded, and so there's two strands, and one of these strands is used as a template to make a copy of a messenger RNA, and then that messenger RNA is used by the ribosomes as protein-making complex in order to travel along that messenger RNA and make the corresponding protein. And the strand that it travels along is called the sense strand. So an antisense strand is the strand that's going to bind to that sense strand. So it's the complementary strand. It's going to bind through that complementary base pairing. The reason why DNA and RNA can bind to one another, and DNA can bind to DNA and RNA can bind to RNA, is because of this base pairing where you get this complementary pairing where each of the four nucleotides, so these letters, are able to pair up with one another. And so when we talk about the antisense strand, we're talking about, or an antisense oligonucleotide, we're talking about a sequence that is going to specifically bind to the sense strand. So it's going to be able to bind to the messenger RNA, or as we'll see, like the pre-messenger RNA and things. So basically, this is just telling us that we have the sequence that's going to specifically bind to a sequence we're going to try to target. Okay, so that was antisense. What about oligonucleotides? So oligo, this just means few, and nucleotides, these are the building blocks of nucleic acids. And so typically when we think about nucleic acids, we're thinking about DNA and RNA, but there can be modifications to nucleic acids, and this is in fact what we're going to see in the case of these ASOs, is that they're going to get modifications that are going to help stabilize them, as well as modifications that can target them to specific tissues or cell types. They can also be attached to or like conjugated to various things. And so you might be able to use an ASO that's conjugated or like linked to a protein or something like this in order to bring over machinery to specific sequences. But what we're going to talk about mainly is going to be how ASOs can be used to bind to pre-messenger RNAs, to bind to messenger RNAs. So basically, when I was talking, I kind of went through really quickly, basically how you have this gene that gets transcribed into messenger RNA that gets translated into protein. I kind of skipped over the detail is in that when the gene originally gets transcribed, it has regulatory regions called introns, and these introns have to get removed in a process called splicing to give you a mature messenger RNA. And during this splicing process, there can be mutations that cause this process to go wrong, and this can lead to various diseases like spinal muscular dystrophy or spinal muscular atrophy. And ASOs can be designed that can actually bind to mutated pre-mRNAs and alter how they're actually used in order to get them to be spliced correctly. And so we'll see a cool example of this with the drug Spinraza that's used to treat spinal muscular atrophy. And we'll get back to that. But I'm going to start by focusing more on how we can use ASOs to target messenger RNAs, because this is the case where you're probably going to hear the most about, and can be used in the lab as well as in therapeutics, and it's a very growing area of research. And it's also kind of easier to explain than the splicing, and so we'll get back to the splicing later. So if an ASO binds to a messenger RNA, there are a couple of things that can happen. And ASOs can be designed based on kind of like modifying that chemistry in order to promote different things happening. So the two main things that can happen are it can kind of just bind there and block stuff, or it can bind there and actually cause that mRNA to get degraded. And initially, they thought that the main way that these ASOs were going to be able to inhibit the use of these messenger RNAs, they thought that it would just kind of like sit there and block things. And so they would be, you can design them, say, to block the site for the binding of the ribosome. So that complex that has to go and make the travel along the messenger RNA and make the protein. Well, if you were to bind a big old ASO on the site, now the ribosome can't find where it's supposed to start, or it would get blocked along its travel. And so they thought that this was going to be like the key way that these things worked. But then they realized that it was actually working via degradation. So those ASOs were targeting these messenger RNAs, and then those messenger RNAs were actually getting degraded. And the way that they were getting degraded is through an RNase called RNase H. So this is a nuclease. A nuclease is going to be an enzyme, so basically a reaction helper, speeder upper. And what this is going to do is it's going to, and nucleases are going to cleave nucleic acids. Different nucleases have different specificities. Some chew from the ends. Some, in the case that we use in the lab a lot, are like restriction enzymes that bacteria have that recognize really specific sequences. We also saw cases where we have enzymes like DPN1 that recognizes methylated DNA. So there are various specificities for different nucleases. And there's this one called RNase H. And so the one we're talking about most is RNase H1. Basically, what it does is instead of recognizing a specific sequence or something like that, it actually recognizes an RNA DNA hybrid. So these can form in your body in at various times as like processing intermediates and things like this. They can cause some problems if not like taken care of, and so your body has ways to take care of them, including this RNase H1. And what this RNase H does is it's going to recognize a DNA RNA hybrid, and it's not going to cut the DNA, that would be too dangerous. It's going to cut the RNA. And so if you have an ASO that has a DNA region and it binds to a RNA, like your messenger RNA, well, now it's going to be a substrate for RNase H. So basically, it's going to be something that RNase H sees and it's like, ooh, that's an RNA DNA hybrid, and I'm going to cut it. And if the RNase H cuts it, now that mRNA can get degraded, those exonucleases, so those things that chew from the end, can take over, and you can get this sequence degraded. But one problem you might find if you are trying to use this RNase H strategy is that your ASO itself might actually get degraded before it can get to work, or it might not bind tight enough. And so a solution to both of these is what's called a Gapmer ASO. Basically, these have chemically modified nucleic acids on the end, so like 5 or so nucleotides on each end that are going to be chemically modified to increase their affinity, so how strongly they bind to that RNA, to increase their stability, things like this. Now, these modified bases, however, the RNase H isn't gonna like see those. That's not. They're gonna be like, that's not DNA, and they're not going to want to recognize this. But in the middle, in between those regions, you have DNA. And RNase H can see, okay, this is DNA, and I've got it bound to RNA, so I've got to cut. I'm going to cut here. And so it's going to cut here. So you design this kind of like window where you have this DNA. This is matching the part that you actually want to cut if you're trying to cut it. And then the ends would, if you were like trying to cut it at a specific place or something, and then the ends would be chemically modified. Typically, you're not really caring exactly where it cuts. And so what people often do is they actually do this like Gapmer walking. So for reasons that aren't totally understood, there are certain places in the RNA that are going to be better substrate, so better able to be cut by RNase H, and you can't just like predict it right out. So what people will do is they actually like will make Gapmer ASOs or make ASOs in that kind of go along a sequence in like overlapping footprints, and they'll test them all and they'll see which one works the best. So they do this like walking And so you might have an ASO that binds here, and an ASO that binds here, and an ASO that binds here, and an ASO that binds here. Basically, you're just trying to find the place that's going to get cut most effectively. And so it can take a lot of work, but it can also make it so that you can find really great sequences and really great sites that can get cut by RNase H. Sometimes you don't want the sequence to be cut by RNase H. And so when we talk about wanting to do things like the splice site modulation, which I'll talk about later, if we're trying to do something where we're just trying to block a site, then we wouldn't want that site to be cut. And so instead, we can make those modifications that RNase H doesn't like throughout the whole ASO. But if you want it to be cut, then you're going to want to have some like normal DNA that can get cut. So let's talk about some of the modifications that we can use. Often modifications are used in order to increase stability. So how like stable it is in the body. Can it survive and stay alive? Is it still active, or is it getting degraded? Such as how we talked about, that could be a problem with the ASOs, is that they could get degraded because you have this like this single-stranded nucleic acid in your body, which isn't very stable usually. So the modifications can help make it stable. They can also improve the bioavailability. So it's basically like how able is it to actually get where it needs to be if you give it pharmaceutically. And so basically, if you were to give this as a drug, would it actually make it to the cells that you need to get it to? Or would it be kind of just like bound up in various like blood proteins and things like this and not be very useful? Or just get like excreted and things like that. And so bioavailability is a big thing with like any kind of drug you're using, and various modifications can make it more bioavailable. So modifications can basically chemically stabilize them. They can keep them from getting cleaved by other nucleases or by the RNase H when we didn't want that, and protect them from being seen as foreign and like attacked by your immune system. Common modifications include modifications to the 2' site. So this is the site where RNA has an OH and DNA has just a hydrogen. That's where you get the deoxynucleotide, is that they don't have the oxygen there. As well as modifications to the backbone, so like the linkages between the nucleotides. And commonly, these 2' modifications include things like a 2' O-methyl. So instead of having an OH, you have an OCH3, so a carbon attached to 3 hydrogens, or you have a fluorine. And so basically, you can have these different modifications to the 2' position in order to better stabilize things. And you can also modify the backbone, such as having and swapping out the phosphor in this normal phosphodiester linkage for a sulfur, which gives you a phosphor thioate linkage. One reason these modifications can help is that basically that OH, that 2' OH that we talked about, it can kind of attack the phosphodiester bond in order to break an RNA chain. And so this can cause your RNA to break. And so if you were to modify that OH so it can't be all attacky, then you're not going to get that chain broken. And if you make the backbone so that it's harder to attack, or it's not attackable, then you're going to stabilize the nucleic acid. And so that's a strategy that can be employed in order to help modify and protect these. An example of this we'll look at later is in Spinraza, that drug for SMA that we'll talk about. It basically has these phosphorothioate linkages that you can see. And Spinraza is going to have actually at this 2' position, it has this methoxyethyl. So that's another group that will be more protective. Okay, and so yeah, so we're going to look more in that into Spinraza later, but basically it binds to a sequence and that normally causes an exon to get skipped and causes it to be included instead, making a functional protein that can compensate for a protein that patients with SMA don't have. Other types of modifications can be used to target these ASOs to specific tissues or cell types. Often what you see is targeting to the liver. And so the liver is kind of a useful target because you can basically conjugate or attach a sugar called GalNAc to the nucleic acid, and then that will bind to receptors on liver cells, so these hepatocyte receptors, and it will allow those liver cells to take up the compound. And so a lot of the ASOs in development and things like this are for liver diseases. So basically, GalNAc is N-acetyl-D-galactosamine. Sometimes you see like 3 of these conjugated, so like attached to a nucleic acid to get more binding. And GalNAc is going to be a binding partner or ligand for these things called Asioglycoprotein receptors, and so ASGPRs, which are abundantly expressed in livers on the surface of liver cells, but not other cells, so you can target them to the liver specifically. And so what happens is that when the GalNAc binds to one of these receptors, it's going to get taken in through something called clathrin-mediated endocytosis. So basically, on the surface of these cells, there's this receptor, and this receptor's job is normally to bind to and remove glycoproteins from the bloodstream. So basically, proteins that have glycans, so sugar chains attached to them. And so these receptors on the surface, they're there because they can then like clean up glycoproteins from the bloodstream. And so you can kind of hijack their system and put a GalNAc conjugation onto your nucleic acid, onto your ASO. Now that's going to bind to the receptor. And now the receptor, what it's going to do is this kind of like the membrane is going to sink itself in and kind of pinch in with that receptor inside. And this is going to allow the ASO a route inside of the cell. There's still other hurdles to trying to get nucleotides into cells, and like even once they're in the cells, like getting out of endosomes and things like this. A good review article, if you want to see what the current state of things is. I found this recent review article you might find helpful. Basically, there are various strategies that are being looked into in terms of like lipid nanoparticles, cation-based complexes. So basically, those nucleic acids are negatively charged, so you can use positively charged polymers, so these long kind of just like positively charged molecules that can kind of bind to and kind of neutralize those nucleic acids, allow them to get in, things like this. Various cool nanostructure things that are a hot topic. So you can see like various ones in development as well as in actual use that use these various strategies. And so I'll post the link to this review if you want to check that out. But for now, I want to go back and talk more about the splicing regulation. So remember, splicing is that process in which we're removing the introns from the pre-mRNA in order to form that mature messenger RNA. So the introns aren't needed by the ribosomes. They would just confuse the ribosomes. The ribosomes would see them as gibberish. So you want to make sure you don't include the introns, but you want to make sure that you include the exons, or at least the ones you want. I'm not going to go into the details here, but basically you can use your cells use alternative splicing in order to sometimes piece together different exons. But sometimes they accidentally skip over an exon that's important, or they leave in an intron, or they splice incorrectly where they use like part of an exon. And this can happen if there are mutations in the sequences that normally get recognized by the spliceosome, so by the complex that does the splicing. If there are mutations at the sites that it recognizes, or there are mutations that cause it to recognize a site that shouldn't be a site, then you can have problems and you can have diseases. And so ASOs can be designed that target those specific mutations in order to try to fix it. One caveat is that when you're trying to design an ASO that's going to target a specific mutation, this means that you would have to make the ASO designer made specifically for each different mutation, which is, if you have something that's a very common mutation, would be less of a big deal. But if you have something that's a rare mutation and you have to make it individually for each patient, this causes both production problems as well as trial issues and things like this. And so I'm not going to get into those details, but basically it can get complicated. But in the case of Spinraza, what it's actually going to do is it's going to target a sequence that most people actually have, because basically the sequence is in a gene that's not the gene that's mutated in the disease. It's a little bit complicated, so let me walk you through it, and then I have a whole video on this. Basically, SMA is caused by mutations in a gene called SMN1 that makes a really important protein, SMN. And this protein can also be made by SMN2, which is another gene that everybody has. And patients can't make SMN from SMN1, but they can still make it from SMN2. But SMN2 doesn't normally get used because there is a single letter difference in the gene that causes it to be spliced incorrectly, making a protein that is defective and can't be used. And that happens in everybody. So in everybody, even patients without SMA, they don't make much of any SMN from SMN2. So most people are making it from SMN1, but with these patients, they have a mutation in SMN1, so they can't use that gene. But you can correct that defect in the SMN2 gene that was causing it to get processed incorrectly. If you cause it to get processed correctly, then you can allow them to make SMN that can then compensate for having that mutation in SMN1. And so that the difference in SMN2 versus SMN1 is going to be a difference that's going to cause the exclusion or the skipping of exon 7. So the protein, the mRNA, is going to be missing a piece. And so if we can promote the correct splicing so that it includes the exon 7, then we can actually get it to make SMN. So how this Spinraza works, this nusinersen works, is it's an antisense oligonucleotide that binds to and hides this site next to exon 7 that normally causes exon 7 to get skipped. So there's this suppression intronic splicing silencer, this ISS sequence that is next to the splice, that's like kind of hiding the splice site because it makes it so that a protein called the splicing repressor binds to that sequence. And if Spinraza binds to that sequence first, now that repressor can't bind, and so now the splicing isn't going to be repressed, so this sequence is going to get spliced here, which allows for the inclusion of exon 7, and this corrects for that defect, and now these patients can make SMN from SMN2 and therefore have a functional protein. And so this is a groundbreaking discovery using ASOs. Just to put things in context, so basically I've talked about various strategies over the years, I guess, that can target molecules at multiple levels. So you can target the gene with like gene editing with like CRISPR-Cas. You can target the pre-mRNA, as we saw today, with antisense oligonucleotides. You can alter the splicing and stuff. You can also target the mRNA with antisense oligonucleotides, blocking its usage by the ribosome, say, and more importantly, triggering its destruction by RNase H. Another thing that targets the messenger RNA is RNA interference, or RNAi, such as with small interfering RNA, so siRNA, or short hairpin RNA, shRNA. In the case of these, you're also using RNA that's going to specifically bind to a sequence. So you are using like an antisense RNA, but how it works is a totally different mechanism. It binds to a protein called Argonaute or AGO, and then it kind of like directs AGO to that site. And then AGO is going to cut the site and chew it up. And so that's a very different than what we talk about typically when we're talking about ASOs, are these short chemically modified nucleic acids that are going to bind to those sites and block things or cause them to get cleaved by RNase H, not by Argonaute. And then we can also target the protein with things like PROTACs, which will tag the protein for degradation, antibodies that will block binding, and functional inhibitors that will do things like block an enzyme's activity. So hope that helped you understand the role of antisense oligonucleotides in all of this. And yeah, keep an eye on you.